study on the neurocystis, which is
I won't be closing the lab today until about 1:30. I've got a meeting until then, so I'll come back and get started on setting up the lab exam. I opened the door to room 218, which is the one right across the hall. It says Student Research Lab above it. If you want anything, slides or anything, put them in there. And I'll put a few microscopes in there. So if you need to do any last minute reviewing the slides, that'll be open. I'll leave a note on the door to security not to lock it, so it should be open through the evening. When you come in, there will be stations set up at the tables, and there will be an answer sheet for you to write your answers down on. I will have up on the board some ones I want you to do first. They probably will include the ocular micrometer standardization, which will be over there, the hanging drop, which is over here, the gram stain, which is over here. If you want to go ahead and get started on your gram stain slide, you can get it made and then let it start drying. And then there's going to be the streak plate over here. Okay, so those are ones I want you to kind of concentrate on first. And then you can go to the other ones as you see fit and as they're available. You can go back and forth to stations. I just don't want more than one station at one person at one station, unless there are multiple setups. So there'll be a couple of microscopes set up for hanging drop. There'll be 2 or 3 set up for standardization, 2 or 3 stations for making gram stain. When you finish that, there will be then some just regular questions, kind of similar to the ones that are at the back of your lab manual. And those will be on separate sheets, and you'll write your answers on those. And then you'll put everything up here when you're done. And so if you can come in, some of you can come in at 8, and then we can start kind of staggering things. But it really typically has taken 2 hours, okay? But if everybody comes in at 9, we're going to be kind of backed up and waiting for things to become available. So any questions about lab exam tomorrow? Okay, we started talking about bacterial resistance, antibiotic resistance, or antimicrobial resistance. To give you an idea of how serious a problem this is, about 15,000 people die annually from infections that do not respond to any antibiotics. That's pretty scary. Particularly if you have to be hospitalized. If you have to be hospitalized, hospitals are kind of a magnet for pathogens. A lot of sick people there. You have a chance of obtaining what for years was called a nosocomial infection. Now there is a new term that's cropping up to replace that term, hospital-acquired infection. So if you see reference to an HAI, that's a hospital-acquired infection. That's something you get when you go to the hospital. Okay? If you are fortunate enough to get those infections, it's estimated that about 60% of those infections are resistant to at least one antibiotic. So even if they do diagnose you, the initial treatment might not work. These can be deadly and lethal, obviously, but even if they aren't, they add a tremendous burden to our healthcare system. It adds about $10 billion annually to healthcare costs to treat these hospital-acquired infections, particularly the resistant ones. So this is a real problem. We talked at the end of the period about how antibiotic resistance is really evolution in action, and it really is natural selection. The antibiotic, or the antimicrobial drug, is the selective agent. That is the factor in the environment that is favoring those bacteria that have developed mutations that allow them to become resistant. Now, what's doubly scary about this is that not only can these mutations arise in one species of bacteria, but they can then easily spread from one bacterium to another. So a resistant bacteria can exchange genes with a non-resistant form and transform it into the resistant form. This particular slide up here shows you some of the mechanisms by which that gene transfer can take place. One of them you are familiar with, transformation. You transformed bacteria with the Piglet gene. If we've got a resistant cell and it's got resistance genes, if that cell dies and releases its DNA, fragments of that DNA can be taken up by a non-resistant cell, and that cell can become transformed by those genes. So transformation is one mechanism of transfer of resistance. But there are others. Another method is conjugation. Conjugation involves the formation of a direct connection, the conjugation tube, or sex pilus, when we talk about piling. And during conjugation, plasmids are transferred from the resistant cell to the sensitive cell, transforming that cell. So the plasmid has the resistance gene is copied, and that copied plasmid moves through the conjugation tube and changes that cell into a resistant one. It's pretty common for antibiotic resistance genes to be found on plasmids. That's where you most likely find them. And the plasmids that carry resistance genes are usually designated as being R plasmids, R standing for resistance. There is a third way that resistance can be transferred, and that is through the phenomena of transduction. In transduction, there is a viral vector that transfers the resistance genes from one cell to another. So it picks it up from one cell and then infects another cell and transforms that cell. This slide just gives you some documented examples of demonstrated spread of resistance genes from one species of bacteria to another. Notice that E. coli, for example, can acquire and donate resistance genes to and from a number of different kinds of cells. Why is that alarming? Why is E. coli particularly alarming? Okay, well it can, yeah, but why is it a concern to you?
It's everywhere.
It's everywhere, and particularly E. coli is one of those gram-negative enteric gut bacteria. So we all carry around E. coli pretty routinely. So if these E. coli can conjugate or be transformed by resistant species of bacteria, all of a sudden we can have resistant E. coli, and then they can transfer those genes to other bacteria. What's also interesting is that gene transfer can occur between E. coli, gram-positive or gram-negative, remember? E. coli is gram-negative, okay, short gram-negative rod. And bacillus. Is bacillus or staph gram-positive or gram-negative? Gram-positive, okay. So we can even have conjugation between very different microorganisms. Gram-positives can conjugate with gram-negatives and exchange genes.
Even with the thick layers, they can make it.
Yep, they can. This particular slide gives you some of the examples of mechanisms of antibiotic resistance. The first one we've already talked about a little bit, and that is the beta-lactamase enzyme. Through mutation, bacteria have produced this enzyme, beta-lactamase, which can hydrolyze the beta-lactam ring. and therefore inactivate those antibiotics, which would include, what are the beta-lactam antibiotics? Two major categories. Penicillins and cephalosporins. Okay? So here is penicillin with its intact beta-lactam ring. And here is inactive penicillin where that beta-lactam ring has been hydrolyzed by the beta-lactamase. Beta-lactamase is sometimes also referred to as penicillinase. That was kind of its old name. So that's one possible mechanism. Another possible mechanism is that drugs to affect cells, particularly if they're going to inhibit protein synthesis, have to be able to get into the cell. So if there are mutations that result in altered permeability of those cells, then the antibiotic won't affect them. That could involve the mutation in a gene for a receptor that the antibiotic would normally bind to. Because the first step in something getting into a cell is binding to its surface. So some kind of an alteration in a receptor. Another behavior that some organisms have evolved is they have evolved drug pumps that actually pump the drug out of the cell once it's empty. Some of these drug pumps can actually pump multiple kinds of drugs. So it's okay, they enter the cell, but immediately the cell pumps them back out again. One of the first documented cases of the activation of a drug pump was the development of tetracycline resistance. So bacteria that figured out a way to get this stuff out of the cell, even though it is able to enter. Particularly for drugs that bind to ribosomes, there are several mutations that can occur in bacterial genes that change the structure of ribosomal RNA proteins and prevent those drugs from binding to the ribosome. Remember that several of those protein synthesis inhibiting antibiotics bind to either the 30S subunit or the 50S subunit and maybe block elongation or distort the ribosome. But if you can't bind to the ribosome, those drugs have no effect. So slight mutations in ribosomal RNA proteins can be an effective way. We talked a little bit about not antibiotics, but sulfa drugs, how some sulfa drugs inhibit metabolic pathways. Particularly, we talked about how the sulfa drug inhibited the biosynthesis of tetrahydrofolate, which is a coenzyme that is needed to make nucleic acids and amino acids. If we inhibit that particular pathway, that really brings that metabolic sequence to a stop and can kill the bacteria. So what do the bacteria do? They develop a metabolic shunt. And through mutations in some of the other enzymes, they are able to bypass that inhibited step and still form the final product. It's very analogous to what happens in a coronary artery bypass. Okay, you've got a blocked vessel. What do? You reroute around that blockage. Here, though, it is a metabolic rerouting that's occurring. Some interesting new mechanisms have been discovered just pretty recently, and one of them is the first documented case of a mutation occurring that protects a drug target molecule, something that the antibiotic is trying to bind to and inhibit. Do you remember what the quinolines do, the fluoroquinolines do? Nope. They inhibit the enzyme DNA gyrase. That enzyme is responsible for the proper coiling and uncoiling of DNA during replication. Cipro is a fluoroquinoline.
Is that also a topoisomerase?
Pardon me?
A gyrase, is that a topoisomerase?
No, that's a different enzyme.
Oh.
Yep. Gyrase and helicase is sometimes called the same enzyme.
Okay.
Okay. So if that drug can't bind to the DNA, it's not effective. there's a mutation that's occurred in some bacteria, particularly in tuberculosis, where they produce a protein designated MFP, and that actually binds to the DNA gyrase enzyme and protects it from binding to the fluoroquinolone. So the antibody can no longer bind to the enzyme that it's intended to inhibit.
Can you repeat what you just said? Sorry.
Sure.
What, like the MFP does?
Okay, so a mutation has occurred in these TB bacteria, and they start producing this protein MFP. And that binds to the their DNA gyrase enzyme, and kind of shields it from the antibiotic attaching to it. Okay?
That's just kind of like a competitive inhibition.
It is kind of, yeah, it is. Yep. But the binding of this protein doesn't affect the DNA gyrase. It still works. The binding of the fluoroquinolone, the antibody, inhibits it. Okay? Some bacteria will just kind of lapse into dormancy when they're exposed to antibiotics. Or maybe they will be triggered into forming endospores and kind of wait till the drug's metabolized before they re-germinate their endospores. And if you are an L-form bacteria, anybody remember L-form from the first exam? They lose their cell wall during part of their life cycle. And some bacteria have developed mutations that allow them to kind of lapse into that L-form stage. Well, if you don't have a cell wall, then you're not particularly sensitive to at least penicillin and cephalosporin type antibiotics. So there's a number of interesting mutations that have occurred that have resulted in resistance to antimicrobial drugs. And as we develop new drugs, bacteria will adapt. And they will adapt new strategies. Why are bacteria able to evolve so fast? What's something that's really in their favor? They replicate really fast. Okay? Short generation time. So, is it in our best interest to try to minimize antibiotic resistance? Absolutely. Where do we start? What would probably be the most effective strategy to try to minimize antibiotic resistance?
Slow down the replication rate.
Okay, but can we really control the replication of bacteria? It might be tough. Nicole?
DNA, attack the DNA somehow.
What public health strategy could we use?
Don't prescribe as many.
Don't prescribe as many antibiotics, okay? The fewer antibiotics that are present in the environment, then the less natural selection that will be occurring. Both doctors and patients are complicit in the antibiotic resistance problem right now. And both doctors and patients need to be better educated about this. Why are patients complicit? Because when they go into the doctor, suffering from some symptoms that make them uncomfortable, or their children are suffering, which is probably even worse, they want action. Okay? However, many of the symptoms that people are trying to address when they go for a doctor visit could potentially be either bacterial or viral in origin. Ear infections are a very common example. So are throat infections. People think that their child might have strep, for example. So what do? You go in and you want relief. You've been up all night. You know, your child's been crying. You haven't gotten any sleep. Doc, I need some relief. Okay? So what do? You pressure them for antibiotics. Okay? Doctors are running people through their office, pretty fast to try to keep up. And what's the easiest thing to do? Write a script for antibiotics. But if it's a viral infection, that's a waste of time, and that will encourage the development of antibiotic bacterial resistance. So overuse of antibiotics is a huge problem. There is another place that antibiotics are being overused, and that is they are being put in animal feed. Anybody know why growers put antibiotics in animal feed?
So they like don't get sick and they can produce more.
Yeah, they really do believe that it increases growth rate of their animals because they have fewer infections. And they put on weight faster.
And then they do it for pigs because of the density. Like if one gets sick, they're all gonna get sick.
Yep, so they're using them for prophylactic or preventative reasons as well. This is really causing a problem in contributing to the antibiotic resistance phenomenon that's occurring. A lot of legislation has been passed in the U.S. within the last decade trying to ban and minimize this. But that might help in our situation, but it's pretty much unregulated in many foreign countries. Okay, so if we ban it, big deal. People in other countries do it and then import their beef or their poultry, and we still have the problem. That's also the case for availability of antibiotics. In many foreign countries, things that are regulated by prescription here can be obtained over the counter. We took a group of students to Belize several years ago, and in the airport in Belize, there were antibiotic dispensing machines, just like a vending machine. And you could put in your money and buy, penicillin or tetracycline or whatever. In India, where TB is a real problem, that isoniazid that I talked about, the antibiotic that inhibited mycolic acid synthesis, is sold over the counter as a lung pill. And so the resistance to that particular antibiotic is really accelerating very dramatically. We've learned a lot. We used to put antibiotics routinely in things like toothpaste and chewing gum. The idea was we were hoping we could just wipe all pathogens from the face of the earth. Farmers tried a similar strategy with DDT. And DDT, organisms developed resistance to DDT just the way bacteria now are developing resistance to antibiotics. There are some other problems. We tend to really like, and doctors tend to like, and pharmaceutical companies tend to like, broad spectrum antibiotics. Those that kill both gram-positive and gram-negative microorganisms. But if you do a broad spectrum antibiotic, then you are increasingly running the risk of a whole range of microorganisms becoming resistant. Ideally, we would use narrow spectrum antibiotics. Ideally, we would use narrow spectrum antibiotics that would have specific targets for specific species. So if you went in and had a strep infection, then ideally there would be a drug that would affect only Streptococcus bacteria and would affect no others. It used to be fairly common when you went into the doctor's office complaining of strep throat to have a culture taken. Because the culture could tell the doctor whether the infection was viral or whether it was bacterial. What's the problem with having to take a culture to get that information? Yeah, it takes 24 to 48 hours to get the results, and in the meantime, you're not getting any antibiotic if you indeed did have a bacterial infection. And there's a risk, we'll talk about later, of not properly treating bacterial infections like strep throat. It can lead into things like rheumatic fever. Okay, we'll talk about why that's the case later on. Well, as doctors got busier, they really stopped taking cultures pretty routinely. And now, probably more than half of the people who go to a doctor's visit never get a culture. The doctor just looks at the throat and says it's inflamed. It might be viral, it might be bacterial. I'm going to give you an antibiotic in case it's bacterial. Okay? The other thing that really isn't done hardly at all is if you're going to prescribe an antibiotic, to test susceptibility of the organism to that antibiotic. Okay, if you go in and you do have a strep infection, and the doctor says, I'm going to give you antibiotic X, penicillin, for example. What if your strain is penicillin resistant? Then it's not doing you any good. So susceptibility testing is something that really should be done on a routine basis. That would allow us to prescribe only antibiotics that the microorganisms are sensitive to. Here is one way you can do sensitivity testing for antimicrobials. This particular test is called the Kirby-Bauer test. And you test antimicrobial sensitivity by seeding an agar plate with bacteria, ideally the species of bacteria that you are suspecting is the cause of the infection. So maybe these are strep. And then placing on the surface of that various discs impregnated with antibiotic. You incubate this and then look for zones of inhibition around those discs. And this gives you information about whether the organisms are sensitive to that particular antibiotic. What's this similar to? Something you've done? The mouthwash thing. Okay. Very same principle. Okay. Are there chemicals in those discs that inhibit microbial growth? Can we automatically just look at the size of the zone of inhibition and say the larger the inhibition circle, the more effective the antibiotic is? We can. Why not? I mean, it's killing more bacteria, right?
Could be some of their byproducts might be causing it.
That's possible. There's something more basic, really.
There might have been less bacteria in there.
Yeah, I think we can pretty much assume that if you stir up your culture that you add to the agar, you get a pretty uniform distribution. Possibly evaporation. Some of them might have a higher evaporation rate, or they may be smaller molecules, which would affect what? Their diffusion rate, right? Those particular molecules have to diffuse out of the discs into the agar. And if you have a smaller molecule, then it's going to diffuse farther, and it's going to have a bigger rate. So basically what they do is they have to measure these zones of inhibition and then compare them to data that's been gathered about those particular antibiotics so that you can determine levels of susceptibility. There is another, this just shows you a Kirby-Bauer test. This illustrates something kind of interesting about antibiotics. Here is the zone of inhibition ring right here for this antibiotic. Here is the one here. And in the middle, we've got kind of an expanded zone of inhibition. This is called synergy, where the two antibiotics reinforce one another, and the combination together kills better than individual bacteria do themselves. Here's another test. Stop that blank for a second. This is a way to be quantitative about testing for susceptibility to various antimicrobial agents. This is called the MIC test. MIC stands for minimum inhibitory concentration. And basically what you do is you take a series of bacterial broth tubes and you inoculate them with all the same amount of bacteria to start with. Okay? And then you put antibiotic in each of those tubes, and each one is a serial dilution of the previous one. And so what you find is, what is the minimum concentration that will inhibit all microbial growth? We can see here that this tube is clear, this one is turbid. So this particular one has this particular minimum inhibitory concentration. So that can help give you some information about dosing of the antibiotic as well, if you know its minimum inhibitory concentration. There's another reason to determine minimum inhibitory concentration. MIC is something you need to know if you want to calculate the therapeutic index of a drug. The therapeutic index is the ratio of the toxic dose of the drug to its minimum inhibitory concentration. Remember, every drug pretty much has the potential to be toxic. So through lab testing, you can determine toxic doses of various antimicrobial drugs. So if, for example, your toxic dose was 10 micrograms per mil, and your minimum inhibitory concentration was 9 micrograms per mil, then your therapeutic index would be 1.1. If there was another drug that had the same toxic dose, but its minimum inhibitory concentration was much less, 1 microgram per mil instead of 9, now we've got a therapeutic index of 10. So when prescribing, you should always try to go for the drug that has the highest therapeutic index. That will be the most effective. It will kill with the least amount of toxicity. Here's another way to determine MIC. It's kind of an interesting innovation. This is called the E-test. It's kind of like Kirby Bauer in that it uses the bacterial plates seeded with bacteria. But here we have these little strips which were impregnated with antibiotic. And the antibiotic is present in a gradient. So there's the highest concentration here, the lowest concentration here. So we just lay these various strips which have different antibiotics. This TZ stands for some antibiotic, IP another antibiotic, and you get these zones of inhibition. What's going to be the minimum inhibitory concentration? Right down here where the zone of inhibition stops. Okay? So you can just read it right off of that E-test. So you don't have to do all of those serial dilutions by this particular method. It always amazes me how people come up with these kind of innovations, and who thought of them? You know, how did they come up with that particular idea? It's a very interesting and innovative way to do testing very rapidly. So this is just kind of a summary slide of some of the strategies to try to limit drug resistance to microorganisms. Side effects. Here are a couple of side effects from a couple of antibiotics that you probably have heard of. This one is called black hairy tongue. This one is the discoloration that occurs through tetracycline deposition in tooth enamel. Black hairy tongue is caused by some people taking the antibiotic Flagyl. My wife had Flagyl when she was in the hospital. Unfortunately, didn't get this. The host cells are killed by the antibiotic. And when they lyse, hemoglobin is degraded. And This is basically an accumulation in the fissures of the tongue, a whole bunch of hemoglobin from dead tongue cells. They will regenerate as soon as you stop taking the antibiotic, and so it's more cosmetic, and it probably itches a little bit, and it's kind of unpleasant to look at. But these are a couple of examples of side effects of antibiotics. Pardon me? It will go away when you stop taking the antibiotic. Yep. Fortunately. In general, there are several examples of different types of side effects of antibiotics. Some antibiotics are actually toxic to human cells, and they will kill cells. Two organs that are most susceptible to antibiotic toxicity are the liver and the kidney. Why are liver cells susceptible to antibiotics and toxicity? Kidneys filter, so that's why they're susceptible.
Liver's breaking down.
Liver's metabolizing drugs, okay? And so many times the metabolic byproducts of those antibiotics are what cause cellular toxicity. Sometimes the antibiotics will crystallize in the urethra, for example, causing stones, which are very painful. The most common side effect is GI disruption and oftentimes associated diarrhea. Some antibiotics will irritate the cells lining the GI tract. And as a response to that irritation, those cells will increase peristalsis. What effect will increasing peristalsis have on your GI tract? Okay.
Less absorption.
You'll have less reabsorption of water, particularly in the colon, and so you will get a more watery stool, i.e. diarrhea. Another thing that can happen is that these antibiotics, particularly if they're wide spectrum, will wipe out your normal flora. And remember, your normal flora is protective. There's microbial antagonism occurring. Several of the enterics that are part of your normal flora will produce lactic acid as a metabolic byproduct. And that will keep the GI tract, especially the lower GI tract, slightly acidic. That tends to suppress yeast from growing. Wipe out your normal flora, pH rises a little bit. A secondary yeast infection can then result from that antagonism being eliminated. There's a kind of scary new condition that is popping up called C. diff. Have you ever heard of C. diff before? C. diff causes colitis, which is an inflammation of the colon. And it can become so severe that you can get lesions in the colon and you can get blockages in the colon. About 10 to 20% of people, and as many as 70% of newborns, can carry around Clostridium difficile, that's the species of Clostridium, spores. Clostridium's facultative or anaerobe. Normally, those spores won't germinate because the normal flora suppresses their germination. You wipe out the normal flora, and now C. diff takes over. And it can be tough to treat. Even after you discontinue the antibiotic, it can be tough to get rid of these and restore your normal balance. Anybody? Go ahead.
Is that like really like contagious?
Is it really contagious?
Because when like it's like we go to patients where they have C. diff, like you have to get on the vest.
Really? Okay.
I know when we handled it in the lab, we had to use it
Really? Okay. It appears it is. Yeah, I mean, you might ask about that, okay? Because I'm not sure. Yes, you could be exposed to the endospores. If you could follow up on that, I'd be interested in knowing the answer to that question. Yeah. Anybody ever taken an antibiotic that where you read the warning on the label and it says avoid exposure to sunlight? Yeah, there are some antibiotics that cause photodermatitis, which is an allergic reaction triggered by sunlight. So that can be a side effect of some antibiotics. Some antibiotics, particularly the aminoglycosides like streptomycin, can interfere with some of the cranial nerves. That can cause dizziness, deafness, and other sensory motor effects. So that's something that's kind of a concern. We mentioned allergies as a possible side effects. About 1 in 20 people are allergic to penicillin. That's something you really should know. It's one of the first questions they oftentimes ask you when you go into the doctor is, do you have any allergies, and particularly latex and penicillins they're interested in? Finally, another possible side effect and concern is inhibition of mitochondrial protein synthesis. Why might an antibiotic inhibit mitochondrial protein synthesis?
Because that's the 70s.
Because it has 70S ribosomes. Mitochondria and chloroplasts have the 70S ribosomes. This particularly happens in rapidly dividing cells. Because when a cell is dividing, doesn't it have to replicate its mitochondria so each daughter cell gets some mitochondria? Yeah. Well, if you inhibit protein synthesis in the mitochondria, you can inhibit mitochondrial replication. If you're taking a 7 to 10 day dose of antibiotic, this is not really an issue. Okay. But if you are on long-term antibiotic therapy, remember I said that TB, because the bacteria grow so slow, you can be on a year-long antibiotic, then this starts to become a real concern. One of the places in your body where the cells divide most rapidly is in the bone marrow. What kind of cells do bone marrow produce? They produce red blood cells and white blood cells. So if your red blood cell content starts dropping, what happens? Well, you become anemic, and there's all kinds of attendant consequences of becoming overly anemic. One has to do with less oxygen being transported, you're right. So if you are on long-term antibiotic therapy, they will test routinely hemoglobin or hematocrit. Hematocrit's the percentage of red blood cells that you have. What if you start depressing white blood cell production? What do white blood cells do? Yeah, then you start getting immunocompromised. So antibiotics are pretty selective, but they're not without risk. And when you consider toxicity, and you also consider the evolution of antibiotic resistance, we do really need to be very careful in their prescription, and we haven't done a great job of doing that. There's a lot of improvement that needs to be made. There's a real concern right now that drug companies are not going to be able to keep up with the evolution of antibiotic resistance because it takes so much time to develop new drugs. It takes so much money, and so many of the drugs that make it into trials never make it to market, in part because they have adverse effects. I included a few slides at the end, which are kind of summary slides, which I think are going to be helpful in studying for this particular part of the exam, which just kind of summarize some of the drugs that we've talked about. If there's things listed on these particular slides that we haven't mentioned, don't worry about them, okay? But it's kind of nice to have everything in one place when we talk about particular drugs. It describes their mode of action, their clinical considerations, and their method of resistance, for example, on this slide. So there's some very useful information there. And there's more antimicrobial drugs. We talked about some antiviral drugs. So that's there just for your reference and the way that you might study, start organizing this information for study. So what we're going to talk about after the exam, lab exam on Thursday, is we're going to start talking about the adaptations that microorganisms have evolved to be able to cause infection. So we're going to be talking about the host-parasite relationship. And we're going to concentrate first on the microorganisms and talk about the battery of offensive weapons that they have that allow them to cause infection. And by the time you finish that section, you're going to go, how am I ever healthy? Okay? Then we will turn the tables and we will spend a lot of time talking about the host defenses. And the answer is you're not sick very often because your host defenses are really quite effective. So we'll break the host defenses down and talk about the first line of defense, the second line of defense, and then really dig in fairly deeply into the third line of defense, which is your acquired immunity, and talk about antibodies and lymphocytes and all the weird interactions that take place between those cells. And then we're going to wrap up. It's kind of hard to believe we're already talking about wrapping up. But we'll wrap up at the end and talk about some applications of immunology in diagnosis. And particularly people like John who want to go into medical technology, we'll talk about some clinical applications of things that you would see in a med tech lab for diagnosis. So that's where we're going, down the road. So we will see you, some of you tomorrow at 8, some of you a little bit later. Yeah, I mean, you can come anytime after 8.
I have 2, maybe 3 questions on the lab review questions.
Okay.
Can I ask you 3 minutes?
Yeah, sure.
Did you record today?
Pardon me?
Did you record?
I recorded.
Did you bud?
Okay.